Excellent. Good morning. It's a great opportunity to be able to come here and share the work we've been undertaking at our university. So we're very interested in using liposomes, as many of us in the field are, and we know liposomes are not a new product. They've been around for a while. We've seen about 50,000 publications, sadly not all mine, but I would love to claim it was the case. But from that, we have only had about 14, 15 products actually made it to the market and being used in patients. And one of the real stumbling blocks for us are that liposomes are quite a complicated formulation to take all the way through to production, and they are quite costly. And that's why we've seen the products coming through are mainly for the high-end, more expensive formulations, such as the chemotherapy, when we've seen the most recent thermodox coming through. So liposomes have a range of strengths, but we also have to tackle this disadvantage in their cost. And the products coming through by 2020 they estimate as a field it will be worth about $20 billion, pounds in that respect. You know, the pounds collapse, so hey, so we get that. And we also can see that, for example, in the UK, Acalix, which is Doxil in the US, it costs about £6,000 per patient. So it's something that we have to tackle if we want to make this more affordable and accessible to everybody. And we've also seen the problem with the FDA. We had a shortage of Doxil, and that was due to cost and production. The advantage of that was we did actually see a generic product coming through, and that was fast-tracked through by the FDA, which was very exciting to see we are now moving into that field. We do recognise that there was some issues with this fast-tracked product, but it was great to get our first generic liposome system through. So the thing is, when we all work in it in the lab, we all use Bangham's method of the lipid hydration. It gets us going, it's cheap, it's easy, and we all know how to do it. But really, the reality is scaling that up. And if you're from the Netflix generation, you know, Walter White, he didn't like to scale up with a round bottom flask either. We need to think about how we can get products off the bench into the market easily for us as researchers, because it's all very well saying we have a new formulation, but we need to be able to get it transitioned straight through into the clinic. And obviously, that's why the microfluidics is such an exciting system. So one of our key research areas, and when we got talking to you in one of the conferences a few years ago now, was we wanted to be able to develop cost-effective, high-throughput continuous manufacturing of liposomes based on what we did in the lab without this very costly in-between stages. And we know there's a range of systems that generally at the moment liposomes, we go top-down, we make our liposomes, we break them down smaller and smaller, and we've already heard the issues that can cause with us. We lose drug loading, we can damage lipids, it's expensive, and we have batch-to-batch variation. So really, we were trying to tackle this issue. So the microfluidic system was very exciting, and we have seen this develop over time. So the range of publications has actually caught up with liposomes in that respect. So it's very exciting technology, as we know, and it has great application for liposome systems. So the advantages we know, it's scalable, robust, and we can go continuous. And that really worked for us because we're keen in two areas, both the delivery of drugs, and we do a lot of work in vaccines, and those really have to be cheap and cost-effective. So we basically set out our strategy, 3 areas we wanted to look at. The making of the liposomes as they are. Also, we were looking at putting low solubility drugs in liposomes, so we can put them in the bilayer like solubilisation agents. Same as micelles, but the thing is they hadn't really taken off because liposomes are slightly cost prohibitive. So we needed a cheap way to use liposomes as a solubilisation agent. And the third area we were looking at, obviously we want high solubility drugs within the liposome core, and we've heard how we can exploit that for targeting, and also in some aspects we can look at that for vaccines. So one of the formulations was looked at, and we just had this accepted for publication, and Smear's got a poster on this, is actually double delivery of drugs. So we put 2 drugs, we can put one in the bilayer, and also, if it's low solubility, and then put a high solubility in the aqueous core. So you can get double encapsulation, double delivery. So that's what we were looking at with the microfluidic system, and we're exploiting it in 2 ways, because we have seen you can get higher loading in both aspects using this system. So we chose to standard phosphatidylcholine and cholesterol. At an 8:1 ratio, we scaled down our cholesterol slightly to allow us to force in the drug within the bilayer. So we use glipizide as a model drug because it's lipid soluble, and we put metformin in the aqueous core. And for those that are clinically, you would appreciate those 2 drugs are often used in combination in patients. So that was the idea. We have our liposome. You can take both drugs at the same time, and we wanted to investigate that. So we used the microfluidics and its standard nano-assembler. So we put our gliposide in with the lipids, and we put our metformin in with the aqueous core. And we just co-inject them, and we get lovely liposomes out about 80 nanometers in size. It's a very straightforward method. So the things we needed to look at when we're making liposomes with the system is obviously our flow rate and our flow rate ratio. We optimize those. We use the standard herringbone. But other things, and it's been touched upon, was the solvent choice, because it's not only the lipids we're working with, but it was the solubility of our drug in that lipid. And for somebody that worked on liposomes, and we always use chloroform, you know, it's great, it dissolves everything. Obviously, it's not good for manufacturing and scale-up, and also the microfluidic system tends not to like getting dissolved by chloroform. So we try and avoid that as much as we can. So the choice was, we need a lipid, we can get our drug into, we can get our lipid into, in that respect. And the clinical attributes we're looking for are obviously composition, what we make, does what we put in come out the same, the size, the zeta potential, as we've heard about Malvern, drug loading, and the drug and lipid recovery. So we first of all looked at solvents that were compatible with all the different parameters we were going in, and ethanol and methanol are good ones. And we also heard about the choice of buffers. So we looked at our standard tris, which we use for our adjuvant systems, and PBS, which we would normally use for anything going IV or oral. So we could see that the combination of PBS and methanol was great for this particular liposome formulation. We get very small liposomes, highly reproducible. So that combination we took forward. But that obviously varies depending on our lipid selection. But for our range that we looked at, PC through to DSPC, they were all very happy with methanol and PBS. We also looked at lipid concentration, because as academics, we're very frugal. You might call us tight-fisted, but we tend to look at very low concentrations. But if you do go too low, our variation gets too much, and obviously we're just not giving it good mixing. So once you go about 1 mg per mil lipid, it stabilizes out and it's very happy. And then we looked at lipid recovery using HPLC, ELSD, and this is work from Mariam, and Carla also worked on this. We can see we get good recovery of a lipid. So what we put in, we get back out. And that's important, and obviously, when we characterize our formulations. So we looked at the range of flow rates, flow rate ratio and flow rates. So we can see here we had a full range of flow rates going through 5, 10, and 15. And we've got our solvent to aqueous ratio. And we measured size and polydispersity and zeta potential. And as I think Kazu also used, we tend to find the 3:1 and 5:1 ratio works well for us. And in terms of flow rate, it didn't really matter, as we would expect. So it means you can put it through the system at increasing rates, and it didn't affect our formulation. So that's good, so you can increase your high throughput if required. So basically, it's a bit like Ghostbusters, which is very topical at the moment. It's all about crossing the streams. So if you cross the ratio of the solvent and aqueous, those are the key parameters, and it's really that's where we get it. So the initial lipid concentration we should take into account. We hadn't initially, but that's important. The ratio we mix at, but actually the total flow rate is irrelevant to our formulation. So they're very robust against that, which is excellent for continuous manufacturing. So we had the liposomes, we figured out our ratio, we know our solvents, so we now want to put our drugs in. That's the key thing. So we're now putting in an aqueous and the lipid-soluble drug. So we can see here metformin is our aqueous-soluble drug with microfluidics and liposomes that are around about 50 to 60 nanometers. We are getting 20% loading, which is very high. And if you compare that to the usual sonication extrusion, we were getting about 4-5% maximum. So using microfluidics, we are able to have much higher loading capacity within the aqueous phase, which is great. And then we've obviously got our gliposide, which is bilayer loaded at about 40%. And the bilayer, again, that's lovely and high. If we do it with sonication, it was around about 10%. And we have shown this with other drugs as well. So we've also looked at propofol, and we get about 40% drug loading in the bilayer. So it's great. So using this system, we are forcing a lot more into the liposome formulation. So that is good for delivery. And then when you combine them both together, we get the same drug loading. So you can actually co-load something in the aqueous phase and something in the bilayer at high loading in a one-step process. using the microfluidic system. So for us, that's very exciting because we are really moving ourselves from a system that's straight off the bench and can be scaled up. And that was what we wanted to look at. So a couple of years ago now, it was actually one of our students, Liz, went out and worked with the Nano Precision guys on how we can do the original, looking at scale up. So I know we now have obviously the system in place, but we were looking at, can we paralyze the chips? So we worked with 2 chips or 4, and that was just with injections, and also continuous pumping systems. So we worked with 1 chip right up to 4 chips. So Liz went out to Canada and helped investigate this, and it was to see, can we actually produce these formulations in large bulk? So here we have, so this was formulations. The batch was made at the time we were in Birmingham in England. So you can see this is our liposomes with and without drug, and this is our size. So that was made in England, and then the 2 drivers were made over in the team in Canada. So we get good matching from that. So it scales between labs, and also when we do the continuous manufacturing flow. So you can see liposomes go straight from one system, scaling right up, similar size, same zeta potential as you would expect, and also polydispersity nice and low. So it was working nicely in scalability with the same production parameters. And in terms of drug loading, again, we see a very similar story. So with the single batches we produced at Aston, we were getting lipid recovery and drug loading, you can see here. So we were getting high lipid recovery, above 95%, and good drug loading, about 40%. And this was with propofol, a different bilayer loaded drug. So it didn't matter if we went batch, syringe-driven, or continuous processing, we get similar drug loading across the systems, which was very exciting for us to see. So that's all drug loading, and that's one area. But another area we're working heavily in is developing new vaccines. And this is, again, where microfluidics really comes in to help us in this. So we know vaccines, we know how they make a global impact and help us reduce drug burden if we're protected. But we also know vaccines are not that cheap, and they should be if we want to have a global campaign. So we need to tackle, basically, we need more affordable vaccines. They need to be easy to use and easy to access. So accessible vaccines may have to be deployed at the point of infection. We saw that in some areas, when we're seeing these breakthrough infections. We should be able to go to the area, deploy and make our vaccines. So that's really where we want to be, and we've got a lot of funding from the EU to consider this. So really the nano assembler comes straight into that aspect for us very nicely. So we have a system that should be able to high throughput or scale in high throughput to give us liposome adjuvants. So one of the formulations we've worked on extensively is a cationic lipid formulation, and it's based on 2 lipids which we get from Advanti. So it's the DDA lipid, dimethyldiammonium bromide. We can see it's cationic lipid with head group, and it's combined with an immunostimulator, trehalose dibenate, TDB. And we combine these 2 lipids together into a liposome formulation. And the good thing is we've got a cationic lipid, which is able to give us a strong Th2, which is basically an antibody type response. But we're working a lot on developing a TB vaccine, so we need a Th1 cell-driven response as well. And actually this cationic lipid is well recognised to give us a strong Th1 response. And basically what you have is you have a cationic liposome, and it's the same as we've seen with DNA, RNA. Quite a lot of our subunit antigens are negatively charged, so you've got a cationic liposome, the antigen sticks in the surface, so it's easy to load. So you've basically got a carrier system. So that should be your cationic lipid. But then you add in your second element, trehalose dibenate, TDB, and that actually potentiates the immune response further. So it then gives us a second boost, you might say. So on its own, TDB won't form liposomes, but it can fit within the bilayer nicely. And then so you've got a liposome that carries your antigen, and then the TDB element helps stimulate your immune response further. So it basically engages and stimulates your antigen-presenting cells further to boost that response. So it's a double delivery system. So when we make these liposomes, this was the standard lipid hydration method that we used to do. And you get liposomes about 400 nanometers in size. Because they're very cationic, they tend to be slightly smaller as MLVs. They have a very positive charge, around about 50 millivolts. And because of that positive charge, we have a negatively charged subunit antigen, which we use for against TB. And it loads very easily. So you make your liposomes, you mix it together, and it sticks on the surface. So it's great. So we get high drug loading. And you can see that here. Here's our liposomes. The bilayer's stained red with the liposomes, and we had fluorescently labelled antigen, and it absorbs to the surface of them. So it's very straightforward. And in terms of phase transition, which was nicely introduced for us earlier by Di, we can see that DDA on its own has a high transition, about 46 degrees. When we add in the second TDB, it does reduce the transition slightly, but more importantly, actually, the combination is much more stable as a liposome system. So not only is it an adjuvant, it actually helps stabilise our liposomes, which is nice. So how does this combination work? Well, we've got, as I say, we've got a cationic lipid. It carries our antigen to our antigen-presenting cells. So our liposomes are here. You've got your antigen on the surface. It takes it to our antigen-presenting cells. The TDB element really potentiates your DCs, activates them, and off they go and do a strong immune response. So it's basically delivery and adjuvant system combined. And the subunit antigen we're working on, as I said, is to protect against TB. So tuberculosis is a growing problem. Globally, it's a major issue for us. And now if you're a child born with HIV, we shouldn't be using the standard BCG vaccine that most of us may have had because there is a risk of reversion. So we do need to develop new vaccines that are based in subunit that are safe for us all to have access to. So the liposome system we've been working on has already been shown to give the same protection as BCG Danish. So that's the gold standard that we're asked to work to within the EU to give protection. So the liposome system of DDA, TDB, which was originally developed by Stanton Serum Institute, and they really were the leaders in this area. So we've got a cationic liposome with a subunit antigen absorbed, and in terms of protection from infection within the lungs in a guinea pig model, you can see it's equivalent. So it's a great liposome formulation, and it's a strong adjuvant. And it works mainly because it is a delivery system. So if you inject a subunit antigen, IM, within the muscle, and you track where it goes. So here we go, we've tracked it down. After about half an hour, the antigen starts to get degraded and it's lost from the injection site. So it's just broken down, flushed away. The body can't do anything with it. But if we stick it onto the surface of our liposomes, DDA, TDB, the antigen's on the surface, you inject it IM, the liposomes stay at the site of injection. And that's shown here with the green line. So the liposomes stay there, and after 15 days, we've still got about 75% of our liposomes sitting there. And they are able to hold the subunit antigen at that injection site. So we have a depot, so it sits at the injection site, holding the antigen. In come our antigen-presenting cells, and that's shown here with blue. So we pre-dose with a blue stain that's taken up by antigen-presenting cells. And over time, you see these monocytes and macrophages coming to the injection site, taking up the liposomes and the antigen and being stimulated to give us a strong immune response. And that's shown nicely here. So the combination of the DDA and the TDB hold the liposomes at the injection site and give us a strong immune response to give us protection against infection for TB. And again, these are animal models. So you can see how the liposome works. You can see it's a great system. But as we knew, lipid hydration wasn't going to cut it. So we needed to figure out how we could produce it. So again, we then looked at the microfluidic system. So we would have our DDA and our TDB, our two lipids, and we dissolve them in IPA because they are quite difficult to dissolve in many lipids. And we can have our aqueous buffer. You can either have it without the antigen, because in our case, we're sticking the antigen on the surface, or you can put it inside. But for us, the idea is we want to be able to rapidly produce the liposomes. Meanwhile, if it's a point of a new infection, we're producing the antigen at the same time. We just co-mix and inject. It's very easy. So that was our model. Thing is, we do have to work at over 45 degrees because the DDA and TDB are not so comfortable in those lipids, so we had to keep the temperature up and work above the transition temperature. So one of the things we thought, well, if you are going to put a sensitive antigen in the aqueous phase, we probably want to bring that temperature down. And as was nicely outlined earlier, if we start to put cholesterol in the bilayer, you can get rid of this transition temperature. So we basically added in enough cholesterol to remove our transition and let us work at lower temperatures. So we had our standard formulation that I've been talking about, CAF01, so that's DDA, TDB. This is a standard dose that we'd use, and that was actually went into clinical trials in humans by SSI again. We then doped in more amount of cholesterol, so we increased our cholesterol amount, and we also had a neutral formulation just to compare. So if we look at these formulations, this is all with microfluidics. So we have our standard DDA, TDB, more cholesterol, and our neutral formulation. So the black bars are our liposome size. So you can see at 3:1 and 5:1, we get very small liposomes, which is great. And we can also keep our high zeta potential, which is shown in the dotted line. So that positive charge was vital for us to make the formulation. So using the microfluidics, we're able to make liposomes of a small size and keep that cationic charge. And we were able to mass produce these rapidly. We also knew that that DDA to TDB ratio is very important. So we had to make sure it was locked, and what we put into the system was what we got out. And the DDA and TDB are slightly less soluble in the solvents we were using, so we tracked that down. So we developed a HPLC method using ELSD, and we've just got a paper submitted on that, working with Carla, and she's also got a poster here on that. And basically, we tracked our lipids down. And we had a solvent gradient to run it. And we were able to see that we used the old-fashioned lipid hydration method. We also used high-shear mixing. And using microfluidics, we always get what we put in is what we get out. So it's always above 95%, which is great because these lipids are expensive and we need that ratio locked down. And it didn't matter if we put in cholesterol. Again, we get good lipid recovery in all cases, and even with our neutral formulation. So that was great. We can make the antigens, the adjuvants, and we have everything in place. We also wanted to make sure that we recovered any non-used antigen because it is important. So we set up a TFF system, Purification on chip, with UCL, and it's just a mini tangential flow system shown here. And we've set up continuous batch processing with this liposome system. So we have our microfluidics, our collection, we feed it through the TFF, and we can recirculate and produce our liposomes at manufacturing continuous. That was the system we set up, so it was small scale, but proof of concept. And we can see here we checked our back pressure, so to make sure that we were able to maintain our liposomes but push out any non-incorporated antigen. So we went up to a back pressure around about 60 to 65. If we went above that, we actually saw our liposomes coming out into the retinate, so that was no use for us. And the key thing was to get rid of non-entrapped antigen and also the solvent from the system. So we can see after 3 cycles, we got nearly all removed, so we got 75% of anything not absorbed and 95% of our solvent we removed from the system. So it was a good high throughput, and we're clearing out non-incorporated antigen and any solvent. So basically, we've got our liposomes, we have the DDA-TDB ratio locked, we prove that, we make the system, and we can remove any non-entrapped antigen and any residual solvent from the system in a one-step process. So basically, in summary, we now have a rapid production system, and if you also, we can look at Carla's poster, she's used it for liposomes and nanoparticles with PLGA, so for those that are at the CRS meeting, we have several posters to have a look at using the microfluidics. We have now implemented proof of concept to allow us to do continuous manufacturing of liposomes for drug delivery and antigens, both that side and our nanoparticles. We have scale up, and we can see the industrial applicability very easily moving forward. And just to acknowledge, we've managed to move from here, and this is Liz, who went out to Canada to make them. That was a 500ml bottle. She was very happy with it, so we took the photo. And thank you for your time this morning.
Nice presentation. I have one question. How do you sterilize your liposome?
Oh, so that's a good point. So for these systems, we hadn't built it in, but the plan is we want to get our liposomes down to a small size so we can filter inline system. Because again, the adjuvants we were making were 500 nanometers, and they're very difficult to sterilize. So we had used gamma irradiation and proved they were stable, but that is very expensive again and wasn't going to be applicable for a high throughput vaccine. So we were just going to put in an inline filter for filter sterilization. That's the plan, yeah. So obviously we're still at a university, but the idea is we can produce them by microfluidics, We can filter out the non-entrapped and end sterilise using filtration, or you can put into the start and the end. So you could aseptically fill as well, and the GMP system that has been developed would be highly applicable for that. Yeah.
Cartridge, by the way, is a product dedicated?
Yeah.
Yeah, we can sterilize the cartridges, particularly as we go to GMP, come sterilized for you. And our colleague Ray will talk more about that later today.
Hi, Yvonne.
Hi.
Great presentation. I've also been working with some double encapsulation. And what was very interesting, you were able to recover some extremely small liposomes with a very, very good homogeneity.
Yeah.
I would like for you to comment a little bit more on the step, on the step right after you recover the liposomes, which is this solvent removal.
Yes.
Because even if you use diafiltration or just dialysis or any other method like that, would that be a bottleneck for the high throughput of this process?
Yeah, that's a good point. So that was one of the things. So within the lab, we just use spin columns, or even dialysis. But with dialysis, with drug loading, it's not so good because you might dialysize out your drug. So it was one of the things. So that's why we developed this TFF system. And now it will flow through, and where residual solvent we've checked by gas chromatography is less than 3% using that. We do 3 cycles through the TFF though. So 1 cycle didn't get rid of it for us, but 3 cycles it was straightforward. And we're doing that we can actually concentrate it up as well. I didn't include that data, but we can concentrate up our liposomes we did it to 5 times concentration. So that's good. So it is one of the things that we had to tackle for us to consider how we would get this, large scale.
Thanks for the nice presentation. Maybe I missed this. How did you go about doing the post insertion of the antigen? Is it antigen specific? And also, how does it compare with the routine method of combining with the aqueous phase?
Okay, so for us, we can exploit the electrostatics. So our liposomes are cationic, our antigens anionic. So all we need to do is mix them, and it's easy. But we have Mariam, who's got a poster, has been working on putting protein within the liposome core using microfluidics, and we are we're exploring that in more detail, and we get high loading. So that was one of the things we wanted to make sure we were working at low temperatures with microfluidics, so that we can encapsulate protein in the one step without any worry about heat stress to the protein. So for the vaccines, we don't need to do it at the moment, but we are now working on one-step entrapment. So, and if you've got, I don't know if you're at CRS, but definitely we've got a poster, Mariam Hussein, if you look at that one. That's
Thanks.
Great. Yeah, just quick question about your dual drug. Adding that hydrophobic drug, what does it do to the stability of like the metformin in terms of leakage?
That's a good point.
And what have you done? What kind of things help recover that?
Yes, so to put the drug in the bilayer, we have to take down some of the cholesterol content because cholesterol and the drug compete. So that is a problem for us. So the drug loading in that respect is competitive. So cholesterol versus drug is competitive. When we put both drugs together, and if we've got a poster on that, actually the combination, so Samir Joshi has got a poster on it. The combination, actually, we got increased leakage. So we think probably by putting the drug in the bilayer, when it comes out the liposome, we're destabilising the membrane, and then the metformin's coming out. So we want to explore that a bit more. So it's different from drug alone, but we can continue to optimise that.
Great. One more time for one more question.
That's me. Hi, Yvonne. Brilliant as always, of course. Just a short question also regarding the double loading of 2 different drugs. Have you ever tried to load at the same time something like an siRNA and a cytotoxic drug, which are very different concerning the PCC?
No, we haven't looked at that, but that would be a really nice idea and a good project. So we haven't done it, but I could see that. We've been playing about and toying with the idea of making artificial viruses for vaccines, maybe along that line. So, but we haven't got that far yet. But it's a great idea.
Okay. Let's do it.
Yeah, let's do it.
Great. Well, thank you
Okay,
so much.
fine.
Thank you so much. That was excellent.